Hello students, welcome to EPG Pathshala. Myself Monica Saini, I am from Department of Anthropology, University of Delhi. Today, I am going to discuss on the module DNA fingerprinting, VNTR, that is variable number of tandem repeats, mini satellite and microsatellite under forensic anthropology paper. After studying this module, you will be able to identify the steps to DNA fingerprinting. You will be able to recognize repeated patterns in DNA fingerprinting and explaining the ways by which DNA fingerprint can be used to apply in public domain. Students will be able to match the unknown DNA fingerprints to the recorded or suspected DNA fingerprint in the Bureau of Crime Investigation or in police investigation or in concealing the personal identification. Additionally, this module will let you to match the unknown DNA fingerprints to the recorded or suspected DNA fingerprint in the Bureau of Crime Investigation or in concealing personal identification. DNA, that is deoxyribose nucleic acid. The DNA fingerprinting is a well-known method of identifying criminals. It is considered as a recent trend in personal identification. It is a technique to find out the variations in individuals of a population at DNA level. The technique of DNA fingerprinting was pioneered and perfected by the British geneticist who was Dr. Alec Jeffrey. In 1884, he had developed this process to visually identify DNA that is found between the genes in the hope that it will reveal the markers for inherited disease and lead to early treatment. He had no idea at that time that his new technique would help to solve cases related to identity by comparing DNA. Human DNA prints are known to possess greater specificity. Like every human organism, it is unique in terms of their fingerprints. Likewise, each individual has a unique DNA fingerprint. Unlike conventional fingerprint that occurs only on the fingertips and can be altered by surgery, a DNA fingerprint is the same for the every cell, tissue and organ of a person. It cannot be altered by any known treatment. It represents the blueprint of the individual's entire genomic makeup. The DNA fingerprints, the specific set of DNA fragments produced by cutting the DNA with a specific set of sequence-specific enzymes, and it depends on the exact sequence of subunits that is called nucleotide pairs in the DNA of the individual. Nucleotides are the molecules that make up the DNA. Each nucleotide has a nitrogenous base, a pentose sugar and inorganic phosphate. There are double ring purine bases called adenine and guanine and two single ring pyrimidine, that is cytosine and thymine. The sequence of nucleotide pairs differs in each and every individual. As human genome contains about 3 multiplied by 10 raised to 9 nucleotide pairs, it is extremely difficult that any two individual other than the identical twins will have totally identical DNA fingerprints. DNA fingerprinting, it can be done by various approaches such as DNA fingerprinting with restriction fragment length polymorphism with polymerase chain reaction with amplified length polymorphism and with single tandem repeats. These are the well-known methods of DNA fingerprinting. Now, you will be knowing about the DNA fingerprinting with RFLP, that is restriction fragment length polymorphism. DNA molecules are long double-stranded structure tightly packed in chromosomes within the nucleus of each human cell. Within each DNA strand, number of genes are present which determine the specific characteristics of the individual. The genetic information contained in gene composition of DNA fragments are only 5%, which are known as coding sequences, and the rest 95% do not contain any genetic information, but it possesses the identifiable repetitive sequences of base pairs referred as non-coding sequences. The RFLP analyzes the length of the strand of DNA molecules with repetitive nucleotide by determining a specific pattern of repeat and then cut those into fragments with specific restriction enzymes. These fragments are then separated by electrophoresis, which sorts fragment with respect to length. The segments are then radioactively tagged to produce visual pattern or DNA fingerprint on X-ray film. The drawback of this method is that it requires considerable amount of DNA to sort fragments by length. The diagram is showing you DNA fingerprinting with restriction fragment length polymorphism. The next approach of DNA fingerprinting is with PCR, that is polymerase chain reaction. DNA fingerprinting with PCR is most widely used method worldwide. PCR technique was formulated in 1985 by Carey Mullis. The method is designed to permit selective amplification of a specific target DNA sequence within a heterogeneous mixture of DNA sequences. The purpose of a PCR is to make a huge number of copies of a gene. As a result, it now becomes possible to analyze and characterize DNA fragments found in minute quantities in places like a drop of blood at a crime scene or a cell from an extinct dinosaur. Starting with one original copy, an almost infinite number of copies can be made using PCR. Amplified fragments of DNA can be sequenced, cloned, probed, or sized using electrophoresis. Using PCR, defective genes can be amplified to diagnose any number of illnesses. Genes from pathogens can be amplified to identify them, that is, HIV, human immunodeficiency virus, Salmonella species, etc. Amplified fragments can act as genetic fingerprints. PCR technique requires some prior DNA sequence information from the target sequence. From this information, two DNA oligonucleotide primers or amplifiers of about 15 to 25 nucleotides long and specific for the target sequence are designed. The primers are then added to the denatured template DNA, which binds specifically to complementary DNA sequences at the target sites. In the presence of a suitably heat-stable DNA polymerases and DNA precursors. Primer initiates the synthesis of new DNA strands which are complementary to the individual DNA strand of the target DNA segment. The diagram is showing you DNA fingerprinting with polymerase chain reaction. Next, I'll be describing DNA fingerprinting with amplified fragment length polymorphism. Amplified fragment length polymorphism, or AMP-FLP, is an attractive version of RFLP as it includes relatively less complicated operation and cost-effective procedure. The method uses PCR technique to amplify the mini-satellite loci of the human genome, thus become quicker in recovery than the RFLP. However, there are issues to this method about the bunching of the VNTR sequences, that is, variable number of tandem repeats, which is due to the use of gel in its analysis phase, which ultimately results in misidentification of the DNA fingerprinting. The next approach of DNA fingerprinting is with STR, that is, short tandem repeats. STR is another method of DNA fingerprinting which analyzes DNA fragments for the number of repetitions at 13 specific DNA sites. It usually or actually triggers specific areas that have short mini-satellites, DNA or repeated DNA base pair sequences. STR thus can be able to compute how many times a particular sequence of base pair repeat themselves on a particular location within the strand of DNA. This advantage makes the DNA comparison matching to an almost positive range. Thus, DNA fingerprinting now provides a powerful new tool for establishing identity or known identity in paternity, rape, and assault cases, as well as other identity cases where issue samples might be available linking a criminal to the crime. The DNA fingerprinting approach has become particularly powerful with the recent development of techniques by which small amount of DNA can be extensively amplified in vitro. This allows DNA prints to be made even when only very minute amounts of tissues are available. Next, you'll be studying different principles of DNA fingerprinting. The ideal way to distinguish an individual from other people on Earth would be to describe his or her entire genomic DNA sequence. This idea takes lots of effort, cost, time, and thus clearly not practical. So, we look for genes that are highly polymorphic, that is DNA sequences that have multiple alleles in the human population and therefore different in different individuals. This becomes the principle of DNA fingerprinting, or in other words, DNA fingerprinting works on the principle of polymorphism in DNA sequences. Important to DNA fingerprinting for determining identity are short nucleotide repeats that vary in number from person to person and are inherited. In human genome, about 10% of the entire DNA is formed of repetitive sequences. These sequences are formed of few hundred nucleotides in which a specific sequence of nucleotide bases again and again without interruption and result in the formation of tandem arrangement. Now, you'll be studying mini-satellite DNA and variable number of tandem repeats. DNA fragments can be categorized as exons or coding sequences that contain genetic information and introns or non-coding sequences which don't contribute any genetic information about the development of the individual. These introns, which seem useless, has been found containing repeated sequences of DNA base pairs. These repetitive sequences are referred as mini-satellites. Mini-satellites can be recognized as repetitive DNA fragments that possess 10 to 16 base pairs short series of nucleotide bases. In human genome, mini-satellites are known to occur at more than 1000 locations. The diagram is showing you mini-satellite DNA. Variable number tandem repeats or VNTRs. VNTRs are mini-satellite DNA with varied repetitions of specific sequences. In one individual, the specific sequence of mini-satellite DNA repeats 5 times, whereas in another, it may repeat itself 1000 times. This makes VNTRs ideal for studying polymorphism. For linkage analysis as genetic marker can be implemented as regulators for gene expression and also used for population studies as VNTRs greatly varies among individuals. The VNTRs of two persons may be of the same length and sequence at certain site but vary at other site. Every human being has a unique VNTR sequence. A person's particular VNTR can be determined by performing Southern blotting through a hybridization process with radioactive probes that target specific VNTR sequences of DNA fragments, which will be discussed briefly later. VNTR sequences are inherited genetically and an individual possesses unique VNTR pattern. While analyzing a individual VNTR pattern, the use of more and more VNTR probes result in more distinctive and unique individualized pattern of DNA fingerprint, which will lead to personal identification of individual in all domains of science. The diagram is showing you variable number of tandem repeats. Now you will be studying about microsatellites. Microsatellites are repetitive DNA fragments that possess 2 to 6 base pairs of nucleotide bases. Microsatellites are generally Ca-rich repeat tandems with repeated or variable number of repeats between alleles, which makes it unique, as Ca nucleotide repeats found very frequently in human genomes and are encountered every 1000 base pairs. Microsatellites are ideal as molecular markers for determining disputed paternity, for studying gene duplication or deletion, for recombination mapping, for population genetic studies, as microsatellites possess locus of many alleles. Thus, the progenitor of particular allele or genotypes within pedigree are often be identified. Some of the diseases that are associated with microsatellite include Huntington gene, Fragile X syndrome, Kennedy disease, myotonic dystrophy, Friedreich ataxia. The minisatellite and microsatellite DNA repeat segments do not have any specific function in DNA structure, but their existence is of great importance. Satellite DNAs has crucial role in the construction of genetic maps, isolation of those genes which are responsible for causing human disease. Techniques of DNA fingerprinting would not have been developed without these satellite DNAs. The diagram is showing you microsatellite DNA. Now, I'll be talking about different techniques of DNA fingerprinting. DNA fingerprinting requires only a small amount of tissue like blood or semen or skin cells or even follicle root of the hair. Typically, DNA content of about 100,000 cells or about 1 microgram is sufficient. The procedure of DNA fingerprinting consists of many steps. In the first step, the DNA is extracted from the nuclei of whatever evidence is available. For example, from WBCs in case of blood sample, or from hair follicle cells that cling to the roots of hair that have fallen or have been pulled out, or from spermatozoans in semen sample. Nuclei of the cells are extracted in high-speed refrigerated centrifuge. The diagram is showing you the DNA molecule extraction from blood sample collected from the individual. In the next step, if the content of the DNA is limited, DNA can be amplified by making many copies of it using PCR or polymerase chain reaction. The diagram is simultaneously showing you DNA amplification through polymerase chain reaction technique. In the next step, the DNA sample extracted and amplified is digested by a restriction enzyme such as EcoRI, which functions to cut the DNA into fragments at specific sites. Thus, these enzymes are site recognizing enzymes for restriction fragment length analysis. The number of these sites present in an individual's DNA dictates the number and size of DNA fragments generated by the enzyme. The diagram is showing you example or functioning of a restriction enzyme. In the next step, chopped DNA fragments are introduced and passed through electrophoresis setup containing agarose polymer gel. This is done for separation of DNA fragments with respect to their length. The separated fragments can be visualized by staining them with a dye that fluoresces under UV radiation. In the preceding steps, DNA is in double stranded form, but now double stranded DNA is split into single stranded DNA using alkaline chemicals. These separated DNA sequences are transferred to a nylon or nitrocellulose sheet placed over the gel. This is called southern blotting. The diagram is depicting the southern blotting of DNA molecules. Next, the nylon sheet is then immersed in a bath and probes or markers that are radioactive synthetic DNA segments of known sequences are added to these. The probe target a specific nucleotide sequences which is complementary to variable number of tandem repeat sequences and hybridize them. The image is showing you the process of agarose gel electrophoresis showing the movements of DNA. Finally, X-ray film is exposed to the nylon sheet containing radioactive probes. Dark bands develop at the probe site which look something like barcodes used to identify items at grocery store. The image is showing you X-ray film showing bands of DNA molecules. Southern blotting was named after its inventor Edward M. Southern, who developed this method. It is a well-known technique to detect specific molecules of DNA from a cluster of DNA molecules. It allows investigators to specifically determine the relative molecular weight of a restriction fragment and also permits to estimate relative amount of DNA molecules in the sample. Scientists use this method to identify sequence of sample DNA molecule by probe hybridization. Procedure of southern blotting
performs by extracting DNA molecule from sample. Then it is chopped into small fragments with restriction enzymes. Then the cut DNA fragments transferred into agarose gel and split into single-stranded DNA molecules by alkaline chemicals. These separated fragments are transferred to a nylon sheet. The sheet is then incubated. Sequence of the single-stranded DNA are exposed to known synthetic fluorescent DNA segments and hybridization occurs and can be viewed through autoradiography on the X-ray film. The diagram is showing you overview of the technique of DNA fingerprinting. Now you will be acquainted with different applications of DNA fingerprinting, which include application in forensic domain. DNA fingerprinting and forensic science is basically pertains to intersection of law and science. DNA fingerprinting is highly specific and unique. No two individuals have exactly same DNA fingerprints except identical twins. DNA can be isolated from whatever evidence collected at the scene of crime like blood, hair, skin cells, saliva, semen, and blood tissues. DNA fingerprints thus can be matched or compared with suspected criminal through variable number of tandem repeat prototype. The second application of DNA fingerprinting is in identification of paternity and maternity. A child inherits his or her VNTR sequence from his or her parents. Parent-child VNTR sequence analysis can be used to solve disputed paternity and maternity cases. The sequences are so unique that parental sequences can be reconstructed even if only the child VNTR sequences are known. This information can also help in solving inheritance cases, legal nationality, immigration cases to verify whether a hopeful immigrant is really close relative of already established resident, identity of homicide victim, or adoption cases. The next application is in personal identification. DNA fingerprinting acts as a genetic barcode to identify individuals. It is very reliable technique by which personal identification can be made with higher accuracy. The another application is identification of racial groups. DNA fingerprinting help identify racial groups to rewrite biological evolution. The other applications include diagnosis of inherited disorders such as Huntington disease, sickle cell anemia, thalassemia, cystic fibrosis, hemophilia, and many more. The other applications include development of cures for inherited disorders, detection of AIDS by comparing the bands of HIV RNA with the bands of individual DNA isolated from blood sample, and the DNA fingerprinting can also be used in breeding program. It permits us to determine the genotype or genome of the individual. Breeders conventionally use DNA fingerprinting to assure homozygous or heterozygous dominance. Offspring of the superior genotype are expected to inherit the highly adaptable characters, and DNA fingerprinting allows precise determination of dominant genotype.
So students, now let us summarize. DNA fingerprinting is a well-known method to determine variations among individuals of population at DNA level. The degree of variation is so high that every individual, with exception of identical twins, produce a unique band pattern as every individual has unique set of ordinary fingerprints. It works on the principle of polymorphism in DNA sequences. DNA fingerprinting requires only a small amount of tissue like blood, semen or skin cells or even follicle root of the hair. DNA fingerprinting can be done by various approaches such as DNA fingerprinting with restriction fragment length polymorphism, with polymerase chain reaction, with amplified fragment length polymorphism, and with STRs. These are very well known methods of DNA fingerprinting. DNA fingerprinting is now becoming a powerful new tool for establishing identity or non-identity in paternity, rape, assault cases as well as other identity cases where tissue samples might be available linking a criminal to the crime. The DNA fingerprinting approach has become particularly powerful with the recent development of techniques by which small amount of DNA can be extensively amplified in vitro. This allows DNA prints to be made even when only very minute amounts of tissues are available. Thank you.